At this time, I would like to turn today's program over to Clotilda Tiling, Senior Product Manager in Microbiology at Illumina, who will introduce today's presenter. Clotilda, you may begin.
Thank you, Ryan. I'm happy to present today Gordon Bentley on ultra-deep sequencing in HIV. Gordon Bentley has been involved in the DNA sequencing industry for over 20 years. Early on, he served as a genetic analysis specialist and senior scientist for Applied Biosystems. He managed a high-throughput genotyping and sequencing laboratory in the Bay Area before joining Roche Molecular Systems as a principal scientist for 9.5 years. While at Roche, Gordon played a primary role in the development of next-generation sequencing-based HLA typing and received the 2008 American Histocompatibility and Immunogenetics Scholar Award. In 2010, he began applying ultra-deep sequencing to the study of HIV in the Laboratory of Clinical Virology at UCSF and continues to pursue these studies as a senior research scientist in the laboratory of Dr. Robert Grant at the Gladstone Institute of Virology and Immunology, which is affiliated to UCSF. The results of his studies of HIV have been presented at AGBT, at the Conference of Retroviruses and Opportunistic Infection in 2012, at the International Workshop on HIV and Hepatitis Drug Resistance this year, and today.
Thank you, Clotilda. I appreciate the opportunity to share a little about our experiences sequencing HIV, the human immunodeficiency virus. Here is a little background regarding this virus. HIV is an RNA virus, part of the family of retroviruses. HIV is further classified as a lentivirus, which is a slowly replicating retrovirus. HIV infects human immune cells, T lymphocytes, macrophages, and dendritic cells. It leads to the death of these immune cells, in particular the CD4 cells, and as a result, the syndrome known as acquired immune deficiency syndrome, or AIDS, is a consequence. HIV infection is treated by the administration of antiretroviral drugs, which reduce the plasma viral load of patients and significantly extend their lives. In 2011, it was estimated that over 33 million people were infected with HIV worldwide. The countries of South Africa, Nigeria, and India have the highest populations living with the infection, with a collective total of over 11 million. As I indicated a moment ago, the administration of antiretroviral drugs continues to be the most effective therapy. However, mutations arise in HIV viral RNA, allowing the virus to evade the antiviral drugs. This is the phenomenon of drug resistance. As the schematic shows here, drug-resistant virus can evade the antiviral drugs being administered and succeed in replicating, causing a rebound in the amount of virus produced and increasing the complexity associated with the clinical management of the infection. As you can imagine, HIV genotyping or testing for the presence of drug-resistant virus before treatment begins is now an important part of clinical care. As it turns out, deep sequencing of HIV using next-generation sequencing approaches provides an ideal solution to this need for sensitive HIV genotyping. Through next-generation sequencing, HIV drug-resistant variants, as in the viruses with the mutations that allow the virus to evade the antiretroviral drugs, these variants can be identified in patient samples at frequencies below 1%. In this webinar, those of you who will find value in what I'm going to present for those of you who are sequencing amplicons, just in general, or those of you who are interested in, of course, sequencing amplicons by next-generation sequencing. In addition, this webinar will be of interest to those of you who are interested in sequencing HIV in particular, and of course, particularly as it pertains to the detection of minority drug-resistant variants and viral haplotyping. By viral haplotyping, I mean the identification of populations of virus based on unique signatures of the variants. This latter approach requires long-read sequencing. The core of this presentation will be a discussion of data we generated here at the Gladstone to answer one of the most basic questions, a question that all of us lab scientists ask when faced with adopting a new technology in the lab or simply changing to a different instrument platform. Will it work on our samples? In our case, we compared the performance of the Roche 454 GS FLX Plus and the Illumina MiSeq systems specifically for HIV variant detection. This work was presented as a poster at the most recent international workshop on HIV and hepatitis drug resistance that was held in June of this year in Toronto. The poster was entitled Deep Sequencing of HIV Variant Detection by Illumina MiSeq and Roche 454 GS FLX Plus. So let's talk about the specifics of this comparison. Very, very basic experiment was conducted. Pardon me. The Amplicon library that was sequenced by both systems contained amplicons defining 465 bases of the HIV reverse transcriptase gene, or I will also refer to it as simply HIV-RT, from 48 different HIV specimens. The target region spans HIV-RT amino acids 50 to 203. Now don't worry, I won't make you look at the variant profiles across HIV-RT for all 48 samples. But I will focus on 23 of these samples that were part of this Amplicon library, which are collectively from just 4 individuals, who I will refer to as subjects A, B, C, and D, who are from studies of pre-exposure prophylaxis, or PrEP, who were tracked longitudinally after stopping study drug. Since no presentation regarding sequencing of HIV would be complete without pardon me, presenting a map of the HIV genome, here is that slide. Here's the HIV reverse transcriptase gene. And as I said, I will refer to it most likely going forward as HIV-RT or possibly just RT. This gene encodes the RT enzyme, the reverse transcriptase enzyme that HIV uses to transcribe itself into DNA, which is then integrated into the human genome. In order to do this, it uses deoxynucleotides in the human host cell. Nucleotide, nucleoside, analog, reverse transcriptase inhibitors, also known as NRTIs, are one class of anti-HIV drugs which mimic the deoxynucleotides the HIV virus is trying to use to reverse transcribe itself. When the HIV-RT enzyme selects one of these deoxynucleotide analogs, chain termination takes place and reverse transcription is inhibited. But as I mentioned earlier, drug-resistant mutations arise, and in this case, the drug-resistant mutations that arise make it difficult for the NRTI drugs to bind to HIV-RT and become incorporated, thus preventing the chain termination events that prevent the virus from replicating. Here's a schematic of the HIV-RT region we sequenced. And I've denoted some of the drug-resistant sites identified in this region that are specific to our studies, notably the K65R, K70E, and M184V drug-resistant sites. You can see that in all three cases, these drug-resistant sites, I show the codon sequence that relates to each amino acid position, and you can see that in each case, the change, for instance, at K65R of a lysine to an arginine is a single base change of an A to a G, as are the other two amino acid changes and drug-resistant sites. Also, just to bring this back to the sequencing realm for a moment, for the 454 GS FLX Plus, this target can be sequenced in its entirety on the 454 system in the forward direction or the reverse direction. I did generate over basically 485 base reads, so each read covers the entire target, either in the forward or reverse, or if you choose to do so in both orientations, which is what I did. We have reads spanning the forward and reverse strands of the Amplicon molecules that were used to sequence on the GS FLX Plus. And I have this little reminder here for myself at the bottom just to say that on the MiSeq, we cover the same target region with paired end sequencing. So we get a 250 base read for each molecule in the forward direction that pairs with a 250 base read in the reverse direction with about a 35 base overlap in the middle. So here's a summary of the Amplicon sequencing workflow we use to generate our data. To begin with, HIV RNA was extracted from plasma samples. Initial amplification of the region of the HIV genome we were interested in was accomplished by RT-PCR. These RT-PCR amplicons were used as template for generating the HIV-RT amplicons that were sequenced. Each amplicon was uniquely tagged through PCR with a barcode sequence, an MID, or a MiSeq index sequence. The amplicons were then pooled to form an amplicon library containing 48 uniquely tagged amplicons, which were then sequenced on the two platforms that we're comparing data from today. For those of you who are interested in some of the finer details, as in which kits were used for sequencing on each instrument, we used the GS FLX Plus XLR70 sequencing kit on the GS FLX Plus, and we also used the MiSeq version 2 500 cycle paired end sequencing kit to generate paired 250 base reads. Subsequent analysis of the data from these platforms was accomplished using the CLC Biogenomics Workbench version 6.0.3 at the time. So all data was compared using one analysis platform. Now let's look at some of the data from these sequencing runs, some of the run metrics that all of us next-generation sequencing people love to look at first. This is just a chart in which I'm combining the read distributions for both platforms into one plot, just to show you that we generated 5 million read pairs on the MiSeq that cover our 465 base target region. Those read pairs came from approximately 10 million individual 251 base reads. And by contrast, the GS FLX Plus generated 727,000 reads, which distribute around the 485 base read length position, and that is because the Amplicons that are sequenced on the GS FLX Plus have 10 base barcode sequences at each end, which adds 20 bases to the length of the read. And as far as the quality of the data is concerned, this is again combining the quality distributions for both platforms. 82% of GS FLX Plus reads score greater than or equal to Q30. 91% of MiSeq reads score greater than or equal to Q30. So we're going to start looking at some of the variant frequency data, and we're going to start with subject D, actually. We're going to work backwards from subject D back to subject A. And so we're going to look at the variant frequency data for subject D at the drug-resistant site M184V, which, if you recall from my schematic earlier, is basically at the 3' end of the target region that we're sequencing. There's a single base change of an A to a G that results in the change of a methionine to a valine at this site. So let me familiarize you with these plots. You're going to be seeing 4 of them. Let me just go through them quickly, or let me go through this particular one so that you get an idea of what you're seeing on the remaining ones as well. So the data we'll be reviewing is for a few individuals from PrEP studies who were identified very early as HIV positive. Study drug was then stopped in these individuals as per study protocol. They were then tracked longitudinally after stopping study drug. The topmost chart contains the variant frequency comparisons, meaning the comparisons of the data from the 2 sequencing platforms, the MiSeq and the GS FLX, the MiSeq in red, the GS FLX in blue. And we also add in a little specific PCR data that was also collected for these same specimens independently. The bottommost portion of the chart plots the viral loads in copies per mil of plasma. And you can see down here the timeline, which is weeks since seroconversion or since HIV antibodies were detected in the blood of these individuals. The sort of grayed area here in these plots is the period during which the individual actually was receiving study drug. So the bottom line here is we want to focus on the comparison of the plots of the specimens over this time frame for the variant frequencies obtained from the sequencing data from the 2 sequencing platforms. And as you can see that they both actually track very closely, including they track closely with the allele-specific PCR data. The dotted lines here, by the way, that span across the charts are read depths. So you can see that the bottom blue dotted line represents the read depths for the GS FLX Plus, while the top dotted line represents the read depths for the MiSeq. And you can see how in this particular experiment, where both systems were sequencing the same 48 amplicons, there is quite a wide difference in read depths owing to the significant difference in read outputs from the 2 platforms. To give you an idea of some of the numbers associated with these data points and how closely they are to each other, how concordant the data is, let's look at this time point here, 24 weeks post-seroconversion. And you can see that the frequency of the M184V drug-resistant variant at this site was determined to be 2.47 by the GS FLX Plus, 2.49 by the MiSeq, 2.04 by allele-specific PCR. So very concordant data. And this is the end of the variant frequency spectrum where we want to have the highest degree of accuracy. Going even lower than this, all three methods were pretty concordant, but particularly the GS FLX Plus and the MiSeq, very concordant at this last time point, the GS FLX Plus at 0.42, the MiSeq at 0.53. Let's go on to subject C now, which we're also going to look at the variant frequency profile across the sampling time frame for subject C. And as you can see, once again, the data is very concordant between both systems and with a little specific PCR. And really, that's all I need to say about this particular slide. The point being, again, that concordance between the platforms is very high. We're going to switch to the other end of the target now and look at the drug-resistant site K65R. And the reason for that is that we want to sort of spend a little time on this particular site is that what we found with specimens who were of HIV subtype C on the GS FLX Plus, when we align the sequence reads after sequencing individuals who are subtype C in the HIV-RT region, we find that there are numerous insertions and deletions in the regions surrounding the K65R codon. So once again, this is in subtype C individuals in reverse strand reads. We find that there are numerous insertions and deletions which actually cause the generation of an erroneous variant frequency result. In fact, these deletions typically cause an erroneous elevation in the variant frequency. The same region sequenced by forward reads on the GS FLX is shown in the next panel here. And as you can see, it does not contain the insertions and deletions that are plaguing the reverse strand reads. And so it is for that reason that we report variant frequencies for GS FLX Plus in subtype C individuals at K65R, a lot of conditions here, only with forward strand reads. By contrast, down here at the bottom, this is another alignment showing you MiSeq reads and the K65R region, and you can see there are no insertions and deletions there as well. So we don't exclude any data when reporting K65R variant frequencies from the MiSeq data. But going back to the top here, one of the reasons we feel is responsible for the insertions and deletions around this region of K65R in the reverse strand reads is that there is a homopolymer stretch that flanks this particular site. And as you know, there are issues. There have been some issues in ultra-deep sequencing relating to the accuracy of sequencing in homopolymer stretches. And we feel that this is one of the reasons why there is some difficulty in getting an accurate variant frequency result from the GS FLX Plus in this particular position. The MiSeq does not have this issue relating to homopolymer errors, and so we did not need to remove any data when evaluating K65R variant frequency in MiSeq data or in the forward reads from the GS FLX Plus. So let's look at some of the variant frequencies for the remaining subjects, and we're going to look at specifically K65R and the other variant that I mentioned, K70E. This is subject B, and we're looking down at the low end of the variant frequency range again, below 2%. And as you can see, over the time period of sampling, both platforms were very concordant, about maybe a 0.3% difference between the two, as are the plots for K70E. So I've got both drug-resistant variants plotted in this particular case on the same graph. And just to give you an idea of some of the numbers at this particular time point, 4 weeks post-seroconversion, the GS FLX Plus says that K65R is 1.35%, and that's from forward reads. MiSeq says it's 1.68%. And lastly, subject A, same drug-resistant site, K65R. Again, you can see both platforms are even more concordant in this case, very close to each other across the sampling time frame. Allele-specific PCR is a little bit below both of them, and that could be because allele-specific PCR is missing some of the molecules that contain the K65R variant just by virtue of the manner in which the allele-specific PCR assay is done. And again, to give you an idea of how close they are at this 12-week sampling time point, they are within about 0.2% of each other. And we're going to look further at subject A for a moment. On this slide, I'm going to try to accomplish a couple of things. First is that this is a viral haplotyping example. This is subject A, again looking at all 6 time points from week 0 to week 52 post-seroconversion. And so what you're seeing here are 6 alignment summaries of the data generated from the specimens for subject A. What I mean is that underneath each of these panels, there are 8 to 10,000 sequences represented in this case. And any time that there is a difference in the sequences in this alignment with the reference sequence, a colored bar appears. And if that bar extends all the way to the top, that is an indication that that difference from the reference is there in all the sequences in the alignment. If, by contrast, that bar only extends up to approximately the 60% line, that means only 60% of the sequences below in the alignment contain that difference, and the rest match the reference. And so on. You can see that there are bars, in some cases, of differing heights. The reference sequence here is the consensus of the sequences for the data at baseline or at week 0. So what we want to see in this particular comparison of data is how the virus changes from week 0 over time. And what we're able to do is look at the entire target region and the variance across that target. And as a result, when you see that there are a number of variants showing up at the same variant frequency, that is a viral haplotype. That means that there's a population of virus, particularly I'm looking at this, the data for week 24 here. There are 60% of the virus in this individual has this particular haplotype, which is different from the virus at week 0, while there are some viral virions that have some of the other variants that have shown up in previous time points, and then the remainder match the reference at week 0. So as you can see here, for this individual from week 0 to week 4, a new virus did appear with some variants that we can see all the way across the target region at 100%. Many of those variants disappeared by week 12, while some of them remained. By week 24, there's a totally new viral population in this individual representing 60%. That population increased slightly by week 36, and just slightly over 60%. And then the entire population, actually, that population disappeared, and by week 52, the remaining viral population looks basically like the population that existed at week 4. So this is what is an example of what we are also very interested in, is the tracking of viral haplotypes. And in this case, we've tracked the viral haplotype of this haplotypes of this individual across the sampling time frame, which is a key aspect of this, that you have specimens representing the same individual over a significant length of time. I've got these two bars here to again remind me that the MiSeq data is comprised of two 250-base paired-end reads in this experiment. So I also want to use this slide to show you that the data in the overlap is also very concordant. We've been looking at, in previous slides, data for K65R, which is at this end of the target, K70E very close to it, and M184V at this end of the target. But we really didn't have any drug-resistant sites that were of interest in this particular study for these specimens that are actually in the overlap. But I looked into the data anyway, and I found a variant that was present in this overlap almost directly in the middle of it, that I hope will serve as a representative of how concordant the data is in the overlap region as well in the MiSeq data. The MiSeq for this particular site, which is a F124L phenylalanine 124 to leucine change, which is a T to C change, single base T to C change causing this amino acid change. And in this position right in the middle of the target, the MiSeq suggests that this variant is present at 0.29%, represented by 312 reads. The GS FLX Plus determined that it was present at 0.26%, represented by 26 reads. At the next time point, you can see the data here. So noteworthy is that we're actually, even at the very low end of the variant frequency range, one possible advantage with the MiSeq is that we are able to feel more confident about variant frequencies obtained when we have many reads representing that variant. When the number of reads drops below 50 representing a variant, some people may start to get uncomfortable in whether that number of reads is really can be trusted. But as soon as you get up into the hundreds of reads, it does bring a lot of confidence along with any variant frequency determination. So finally, as far as our observations and conclusions from this comparison, the MiSeq and the GS FLX Plus were highly concordant for variant detection. As I showed you, variant frequencies were obtained from both sequencing platforms, all the way from 100% down to less than 0.5%, and they were also concordant with our allele-specific PCR data. There is a high throughput potential observation here that is associated with the MiSeq when we are seeing concordance obtained with read depths as low as 6,400, and that was a read depth obtained on the GS FLX Plus. That by extrapolation, basically you can come up with the idea that, well, if you don't need 100,000 reads to be confident in your variant frequency identification, then you can run many more specimens, many more amplicons, many more samples, possibly up into the hundreds of samples per MiSeq run, and achieve the same accuracy and confidence in variant frequency identification. However, there are some advantages from having the higher read depth, and one of them is that you can correct for errors associated with oversampling by the use of approaches such as primer ID. And as I just mentioned, having a sufficient read depth to provide additional confidence in identifying variant frequencies at the very low end of the spectrum, below 0.5%, is also a reason to retain as many reads as you can in a particular experiment. So with the primer ID approach, for example, which many of you may know about already, you probably, for example, if you had a 10,000 copy input, these are 10,000 RNA, HIV RNA copies into your initial RT-PCR reaction, you would want to have 30,000 reads from the analysis of that particular specimen in order to, a minimum of 30,000 reads in order to be able to use the primer ID approach and correct for any oversampling taking place there. As many of you know, oversampling can supposedly lead to errors, which can result in variants being identified that are not actual legitimate variants, but may simply be associated with PCR misincorporation errors. Finally, a very important conclusion for us here at the Gladstone working in this field is that this particular experiment once again confirms the low prevalence of minor sequence variants in individuals in studies of pre-exposure prophylaxis, even when analyzed using the high sequence read depths that are possible on the Illumina MiSeq. And with that, I'd like to conclude my presentation and acknowledge the teams that I've been working with, most specifically the team here at the Gladstone Institute of Virology and Immunology, the lab that I'm part of, which is led by Robert Grant, and our close colleague, Dr. Terry Liegler at the Laboratory for Clinical Virology at San Francisco General Hospital, UCSF. Mohammed Mohsen, another colleague who is at UCSF and also at the Blood Sciences Research Institute. We'd like to thank Dr. Sheng Ding of the Gladstone Institute for Cardiovascular Disease, who allowed us to use his MiSeq instrument. And also, I'd like to mention, while not listed here, Dr. Bob Chadwick and the people in the Gladstone Genomics Core. The next group I'd like to acknowledge are the people from Illumina who have allowed us to investigate their technology. Leith Tonkin, who provided invaluable assistance at the beginning of this project in getting us up and running. John Grip, who made the connection between us and everyone who was required to allow us to take this opportunity and make this comparison possible. Clotilde Teeling and Susan Knowles in marketing at Illumina, who made this webinar possible. I'd like to also thank Goli Chariot and the people at Roche 454 for making it possible for us to use a GS FLX Plus to generate the data used in this comparison. And finally, the Gladstone Institutes as a whole for supporting us, the Center for AIDS Research, and the NIH. Thank you once again, and I am ready to take any questions you may have.
Thank you, Gordon. So let's try to actually get as many questions as possible, and please submit them through the Q&A chat. I will ask you a simple one to start with. One person asked, how much plasma was used as an input prior to PCR?
Typically, we use 1 milliliter of plasma. We extract RNA from 1 milliliter of plasma.
Okay.
That would have, some bearing. Viral load does have a bearing on that, and if we need to have more copies going into a particular assay, we may extract from more, but typically it's from a 1 mil aliquot.
Okay. The other question that goes with this one is, what is the mutant viral load in those samples in general?
what is the mutant viral load? So I guess by the question, what you're wanting me to discuss is the difference between wild-type virus and that which is considered mutant.
That's correct. So the percentage, just an approximative percentage of what would be the viral load in there.
Well, the viral load. Well, that's a very difficult question to answer because every individual will have a different distribution of wild type versus mutant virus. And particularly, when people are being infected, the person who gave them the infection, for example, may have had exposure to antiretroviral drugs. Their population of virus in the individual who has infected another may contain wild type plus a number of different mutant strains of virus. So that's a very difficult question to answer, and I think I would probably just go to the slide I showed all of you just a moment ago of this individual, where you can see at week 24, this individual has the virus that it had at week 0, which has mutants, I don't want to use the mutant word, has variants in it. It also has now 60% of the virus has all these other variants compared to the virus present at week 0, as well as almost 80% of the virus contains some of these other variants that are present here. So it's a complete mixture of viral strains. And so the actual proportion of wild type, which has no variants in it at all, is going to be probably very low.
Okay. Okay. No, that's fine. Another question. You looked at mutations, and could this be applied to deletions?
Well, any deletion, okay, it typically will cause a frame shift unless it's being compensated for by an insertion at another position. So if there is a deletion, then typically that is not tolerated. If that is the only deletion, the frame shift will result in an amino acid. Basically, it'll most likely cause a stop or an amino acid sequence that will not result in a viable viral copy. So deletions typically are not
looked at. Okay. Yeah. Another question: Could you describe the pipeline to call the variants?
Oh, sure. Actually, maybe let me back up here in this presentation. So here I described the workflow, and I do realize that I glossed over a lot of the detail involved here. Of course, the proficiency that you employ at this stage of the game, particularly the first 2 steps, how well you do how accurately you can generate your amplicons and how free they are of any non-specific products, will then result in a very clean next-generation sequencing result. And then after that, you have sequence reads that need to be aligned to a reference and variants identified, their variant frequencies called, and the corresponding non-synonymous amino acid changes also recorded. And that is what the CLC Biogenomics Workbench will do for you, all of those things. It will take the sequence reads directly from 454 or from the MiSeq. So in the case of the 454 GS FLX Plus, the sequence read file is the SFF file. For the MiSeq, it is the list of FASTQ files. and in either case, the CLC Genomics Workbench will allow you to do the alignments you need to do and move directly to calling variants by base quality, so by variant quality. So, and then the resulting variants are added. What is added to that table is a list of the non-synonymous amino acid changes. This you can all do by setting up workflows as well, so you don't need to do all those steps manually. You set up a singular workflow, and the genomics, CLC Biogenomics Workbench will actually process through all of your data for you automatically, and you end up with variant frequency tables containing all that information for each specimen that you have sequenced. I don't know if that answers your question, but that's pretty much
it does. I have an interesting one, and it's in your opinion. In your opinion, do you believe the MiSeq is ready for routine resistance testing?
In my opinion, it is a very strong candidate for that application. As far as the workflow is concerned, there aren't any particularly complex steps or procedures that need to be taken to prepare specimens for sequencing on the MiSeq. I do think that making it actually ready for use in a clinical setting is going to require that consortiums be put together, groups of people who have the same interests and goals in mind, and reproducibility, sensitivity experiments will need to be done, and of course setting of cutoffs, appropriate cutoffs for determination of what is a positive variant frequency result versus what would be considered a negative or non-clinically relevant variant frequency result.
Okay. Now I have another one, which is, can you comment on the relative cost per sample in each platform?
I apologize. Could you repeat the question?
Yes. It says, can you comment on the relative cost per sample in each platform?
Okay, that's a very good question. What I can comment just kind of on the top of my head here, and so I will say that emphatically, this is just coming from the top of my head here. I don't have hard numbers in front of me, but if you recall, in this case, we looked at 48 amplicons from 48 different specimens. So you can basically say that we looked at 48 samples on each platform for each run. So on the MiSeq, to generate that data for 48 samples, it costs us approximately $1,500. That's for all the sequencing kits and indexing kits, etc., put together. One run on the MiSeq of, in this case for that Amplicon library, costs us $1,500. So if you want a very quick calculation, if you say, okay, what would it be for 50 samples? Basically, it's $30 per sample, if I do that quickly in my head here. By contrast, on the GS FLX Plus, all the kits to generate the data for the same 48 samples cost about $6,000. So quick calculation, basically that's what, $120 per sample.
Oh. Yep. So we'll do one more to wrap this up, and I will choose this one that says, do you have recommendation for analysis software suitable for handling deep sequencing data?
Well, I think I would, simply because I have experience with the CLC Biogenomics Workbench at this point, which I acquired because it has all of the tools that we who are doing this kind of work need. It was the easiest application to implement here, and as I said, it will take data from multiple platforms, and it has the ability to set up workflows, so it will process through your data automatically. I have to say that from my perspective, as I sit here today, that's the application that I would go forward with until perhaps another application comes available where it might do some more automatic reporting of data. So why did I use that application? Also was because Illumina doesn't have an Amplicon variant analysis software that will accomplish those tasks. But really, that's okay because this application works very well.
Thank you, Gordon, so much for participating and giving this presentation for us.
Thank you,
Ryan.
Thank you again for joining us today. This concludes today's webinar. You may now disconnect.